you have to thoroughly understand membrane potentials in order to understand the physiology of an action potential. Hence the membrane potential iTunes video, and now this one. Okay, so action potentials are accomplished by voltage-gated ion channels, and we're going to designate them as our red square. So we have two of them. We have a voltage-gated ion channel for sodium that is specific for sodium, and we have a voltage-gated ion channel that is specific for potassium. And this cell is at rest. And again, the polarity right here is all around the cell. I've only shown a patch here. Okay? So, in order to fire an action potential, nerve cells express sodium and voltage-gated ion channels that are specific for either sodium or potassium. In other words, these cells have to have a gene to produce these proteins. And they do, okay? Particularly neurons, all along the axon. Because we're really talking about the phenomenon along the axon, okay? There's a different phenomenon that's going on the soma of the nerve. This phenomenon involving voltage-gated sodium channels and potassium channels are only on the axon. Well, the axon hillock too, but we'll get there. So, we have to stimulate the cell, and by stimulating the cell, sodium voltage-gated ion channels open, and sodium rushes down its concentration gradient, as depicted by the arrow. So you tell me, what's that going to do to the voltage inside, or the negativity inside the membrane?
It's going to become less negative.
It's going to become less negative, right. So essentially, in that case, we can reverse the polarity. Okay? Because now sodium is rushing in faster than potassium is leaving, and as a result, more positive ions in terms of sodium are added to the inside of the cell than are lost by potassium leaving, and as a result, the polarity is reversed. So this is only the beginning. This is what we call depolarization. So the reversal of polarity caused by potassium voltage-gated ion channels to open and the sodium voltage-gated ion channels close. What does that mean? That means when we depolarize, we have to put the cell back in its normal state of affairs. That is, at rest, it's negative inside and positive outside. So we have to go back to that so we can fire another action potential. Okay? So, when the polarity is reversed, that's the stimulus. Remember, I said a moment ago that voltage-gated ion channels respond with conformational changes dependent upon the voltage of the membrane. So when the voltage polarity reverses, that's the signal that opens potassium voltage-gated ion channels and closes sodium voltage-gated ion channels. Okay, what is that going to do to the polarity? Make it reversed. It's going to reverse the polarity. Because in this case, potassium is going to be leaving faster than sodium coming in. So again, we're adding and subtracting along the number line. If we're losing a positive charge faster than we're gaining a positive charge, that's going to move the inside of the membrane back towards its negative state. So are you with me? Okay? When the polarity reverses, that's the stimulus that causes the potassium voltage-gated ion channel to close. Now we're back at rest. What happens after that? Well, the sodium-potassium ATPase, or the sodium-potassium pump, reestablishes the ion gradient for sodium and potassium. So we're back at rest. Any questions? Okay, so let's talk about threshold and how the threshold relates to the action potential itself. Threshold is defined as the point at which the sodium and potassium currents are exactly equal and opposite, and an action potential takes place when sodium conductance slightly exceeds potassium conductance across the membrane. At this instant, the membrane is depolarized 10 to 20 millivolts. A weak subthreshold stimulus are not relayed down the axon. So you have to get the threshold to fire an action potential. And every neuron has a threshold that has to be gotten to in order to fire an action potential down the axon. Stimulus at or above threshold always generates an action potential. And the action potential is an all or none phenomenon. Okay? In other words, if you stimulate a nerve cell a lot and it's above threshold, you're going to get the same action potential as if you just get the threshold. The action potentials are going to have the same amplitude or the same deflection from baseline or the same negative polarity reversal from negative inside to positive inside. Okay? So let's take a look at this experiment so we can further describe how this was determined that threshold really exists. And this is a 2-electrode experiment again. Okay, we have a probe inside the cell and a ground in the extracellular fluid, and that is measuring on an oscilloscope the action potential. And then we have this voltage stimulator that essentially voltage clamps the cell by sending a specific measured voltage through the cell that is determined by the experimenter. Okay? And we call that voltage clamping. So in other words, you can change the voltage on the cell, and that will influence what is read on the oscilloscope. So let's voltage clamp, and that's what this represents up here. You have 1, 2, 3 different voltage clamps. Okay? And the first voltage clamp, you obtained this much depolarization, and you didn't fire an action potential. You increase the voltage, that would be the next level, and you got this far from resting membrane potential, or this much deflection. And again, you didn't get the threshold, so therefore you didn't fire an action potential. Okay? Then you give it a little more, and lo and behold, you get to fire an action potential. So the initial clamps, alteration of the membrane voltage, did not get to threshold. They were subthreshold, and you didn't fire an action potential. When the action potential was at or greater than threshold, you got an all or none action potential. Okay? And it doesn't matter if you gave it more voltage. You're going to get this height of the action potential. That's the intensity. And it's not going to get any higher than that. Okay? So that's what we mean by an all or none action potential. So, as mentioned down here, with any additional depolarization, there is no additional amplitude of the action potential. The action potentials are all or none. So let's look at this from a slightly different perspective. Okay? So let me describe this diagram first. So this right here represents the cell membrane. And so does this down here. Now I separated the membrane. I actually lifted this from this and moved it down here. Okay? It's the same exact membrane. I just separated them so I could separate the voltage-gated sodium channel from the voltage-gated potassium channel. Are we okay with this? Okay. So then, this is the outside of the membrane, and this is the inside. And since it's the same membrane, the same is true down here. Now this would be our oscilloscope action potential. Okay? And I added threshold in here. Now when you see an action potential on an oscilloscope, you really don't see the threshold. Okay? I just added it there for clarity. We have to get the threshold to fire an action potential. Any sub-threshold stimulus, and we won't. Okay? So, in addition, the old neurophysiologist, to better describe or to more easily describe the voltage-gated sodium channel, they described it as having two gates, an M gate and an H gate. Okay, now keep in mind, this is not the truth. Okay? This is just a means by which to more easily describe the sodium voltage-gated ion channel. Okay? Because the sodium voltage-gated ion channel has some unique aspects to it. And we'll define what I mean by that in a moment. Whereas the sodium voltage-gated ion channel just has one gate. Okay? As I said a moment ago, they're not really gates. Is what they are different conformations of the protein. This is an integral membrane protein. Okay? So, let's go through this then. At rest, the sodium voltage-gated ion channel is closed. And so is the potassium voltage-gated ion channel. Okay? Now we get the threshold, and we fire an action potential. By getting to threshold, the sodium voltage-gated ion channel opens. So the M gate opens. Okay? Now notice that the H gate was open already at rest. Doesn't really matter. Okay? With the M gate closed, no ions are going to go through. You get to threshold, and the H gate and the M gate are open, and that allows sodium to rush down its concentration gradient into the cell. And that causes the depolarization of the cell membrane. Now, when you get to the height of the action potential, that's the stimulus that closes the H gate and opens the potassium gate. Okay? So by opening the potassium gate, we're going to lose potassium out of the cell faster than we're going to gain sodium. And as a result, we're going to lose positive ions, and that's going to pull the resting membrane potential back towards potassium's Nernst potential, right? And that's exactly what happens. We start to repolarize. Okay? Then, during repolarization, the H gate remains closed, the M gate's closed, potassium gate's open, and potassium's gate, it opens quickly and closes slowly. So what's that going to do? That's going to cause a little bit of hyperpolarization. A little bit of hyperpolarization. In other words, the resting membrane potential is going to move even closer to potassium's Nernst potential. Because the gate's open a little bit too long. Okay? That has some physiologic significance we'll talk about in a moment. And during this hyperpolarization, where the potassium voltage-gated ion channel remains open for a brief time, the H gate opens. Okay? And then, since we're at depolarization, that's the stimulus to eventually close the potassium voltage-gated ion channel. And at rest, the sodium voltage-gated ion channel is closed as well. Okay. Let's go back over this. Let's make some points here. The points are, these gates are not real. It just allows me to easily describe what's taking place. The reality is this integral membrane protein called the sodium voltage-gated ion channel has 1, 2, 3, 4 different conformations dependent upon the voltage of the membrane. That's pretty wild, isn't it? Whereas the potassium voltage-gated ion channel only has 1, 2. And that's all it needs. Are there any questions? Okay. Let's look at this from the point of what we call refractory. There is an absolute refractory period where, what does that mean? It means basically we can't fire an action potential on top of another action. Why? Because the H gate is closed. The sodium voltage-gated ion channel has acquired the conformation that will not respond to stimulus. That's what we mean by absolute refractory. And as a result, we can't fire an action potential on top of an action potential because the H gate's closed. And the H gate does not respond to threshold stimulus and won't open. It's not going to open until we get to baseline. That's the stimulus to open it. And when the H gate opens, then we can fire another action potential.
So it needs to go through the whole process to send another action potential?
Yes, that's one way of looking at it. You got to get the H gate conformation to open. Only the M gate, or the M gate conformation, can respond to threshold. Okay? Okay, let's talk about the relative refractory period. The relative refractory period is when there is hyperpolarization. So the bottom line is hyperpolarization and the relative refractory period are really the same thing. So what makes it relative refractory? It's hyperpolarized. So that means, now check this out, from here to here is threshold, right? Threshold doesn't change. Threshold is all the way across. But when we're hyperpolarized, now we've got to go from this distance, from hyperpolarization, all the way up to threshold, which is more depolarization or more stimulus required than when the cell is at rest, where the threshold from here to here is approximately 10 to 20 millivolts. And now we're something like 20 to 30 millivolts. So we have to stimulate more in order to get to threshold and fire an action potential. We can still fire an action potential during relative refractoriness, but now we have to overcome the distance or the stimulus required, the additional stimulus required to get to threshold. In addition, potassium is leaving out of the cell faster than at rest, right? Because not only are the facilitated diffusion channels continually leaking sodium and potassium across the membrane, they do that even during an action potential. So we have to deal with the excess potassium leaving over and above the facilitated potassium channel. The additive would be the potassium voltage-gated channel closes slowly, and that additional potassium has to be overcome. So you need an additional stimulus to get the threshold. Make sense? Okay. So, the sodium channels contribute to the absolute refractory period for one reason. The sodium gates are closed directly following an action potential as a result of the slow conformational change in the H gates. In other words, some of the H gates are still closed and cannot open in response to stimulus. Or at least the H gate conformation, because we know they're really not real. Okay? So, potassium channels help to produce the relative refractory period for two reasons. During the undershoot of the resting membrane potential, the membrane is hyperpolarized and so requires a larger depolarization to reach threshold. With potassium channels still open, or that is the voltage-gated potassium channels, the resting membrane potential is low or more negative, and a given amount of current will produce a smaller potential change than when the axon is at rest, when it's polarized. In other words, there is more potassium current to overcome. So this is one of the ways in which you can modulate, basically, neuronal output by hyperpolarization at the height of the action potential, right? Or during depolarization. This is even better. During depolarization, remember, ENA is a positive 60. That would be up here on the oscilloscope, right? Right?
Yeah.
Okay, and EM, of course, is a negative 79 millivolts. That would be right there. And EK equals a negative 89 millivolts. Okay, which is down here someplace. Okay, this is the reason why these action potentials are all or none. They're all or none. They're all or none because of the sodium voltage gated ion channel. It opens and then it closes right there. And as a result, you're going to get to that height of the action potential, and you're not going to get any higher, simply because the sodium voltage gated ion channel closed. And obtain the conformation that cannot be stimulated to open. The H gate's closed. Okay? And the potassium voltage gated channel opens right there as well. So there's two reasons why these action potentials are all or none, and the height of the action potential is the same no matter what. The sodium voltage gated channel closes, and the potassium voltage gated channel opens. So it's all or none. The amplitude does not change. Does that make sense? Okay.